complement cascade system, or the complement system, it's not really as difficult as students have a tendency to make it out to be. But there is a lot of jargon in here, and that makes it a little difficult, especially for those people who wait for the last minute to study this stuff. Complement system is usually the hardest chapter for students across the board. And I'm going to try to break it down for you as much as possible. This is the first type, and it's just going to be kind of an introduction to the complement system. Complement is really just a lot of different proteins flowing through the bloodstream, and they're like little hand grenades. They don't do any damage until they break apart, until they blow up. So imagine you're a normal soldier walking through a battlefield, and your enemy soldier walks through the battlefield. These are smart grenades. The smart ones won't blow up near your cells, your surface, but they will blow up near the surface of the foreign soldier. So that's kind of how they work. The whole thing was discovered back in the late 1890s. Jules Bordet from the Pasteur Institute. But you have to understand, a lot of people were doing the same experiments at the same time, or similar experiments. Paul Ehrlich was also doing stuff, and he was in Germany. There was a kind of a scientific revolution occurring around this time. Well, what he noticed is that he could take Vibrio cholera cells and get the bacteria to lyse if he mixed them with anti-serum from an animal that had been inoculated with Vibrio cholera. So the blood serum would lyse the cells. But if he heated up the cells, I can't remember what temperature, I think it was around 55 degrees for a few minutes, then it destroyed the ability of the serum to lyse the Vibrio cholera cells. Now, if you come back and you add back serum from an animal that had not been vaccinated against Vibrio cholera, that had no ability to lyse the cells on its own, and you mixed it with the original serum that had been heat deactivated, then you would get lysis occurring. And he correctly assumed that there were two components involved, one that was heat stable and one that was heat labile, meaning that one was destroyed by the process. Now, Paul Ehrlich was the one to actually coin the term complement, because he said that these other elements are complementing the activity of the first. And eventually they came to recognize these as being proteins. There are a bunch of these proteins. There are at least 30 that are recognized now, and the numbers keep going up. I try not to bog myself down too much in learning all of the individual proteins, but there are a few in here that we'll have to go into. The next little audio clip will be on the functions and components of the complement system.